So we've talked about innate immunity. Now we're going to move on to adaptive defenses, which means defenses that we develop once we've had an exposure to what we call an antigen. The antigens are foreign markers or chemical markers on foreign objects, bacteria, or any one of the things that we've talked about, viruses, etc. If there's a little protein or even a carbohydrate type of marker on them, even if the whole thing doesn't get into us, if just a part of it gets in, like this little part here manages to get into our bodies, okay, that alone, it could act as an antigen, okay, and could sensitize and activate our immune response, okay? So sometimes that's why we have allergies to things that are totally harmless, essentially, too, because sometimes we misunderstand something that is harmless to be an antigen, and we react to it as well. So antigens, again, chemical markers for foreign substances that our body has developed an immune response to. Here we have two different ways of responding. We have cell-mediated, which is through the T cell line. We talked about T cells earlier. Remember, we have the hemopoietic stem cells in the bone marrow, right? They differentiate into multiple different types. Those that differentiate into lymphocytes. And under the influence of interleukin become B cells. Those that migrate to the thymus become T cells. Okay, the T cells have four different types. Cytotoxic T cells, which are really instrumental in our cellular mediated immunity, or like cell-to-cell combat sort of missions. Helper T cells, which we'll talk about more. Regulatory T cells, which help with kind of keeping things from getting out of hand, and memory T cells that hang around and wait till the antigen comes back again and activate the system rapidly. B cells, we call this system humoral mediated or antibody mediated immunity, because these cells are more like the drone operators, let's say, in a military situation. Now, they don't want to actually get out there and risk their own lives, so to speak, right? So lymphocytes do circulate as white blood cells. 10 to 15% of the circulating white blood cells are B cells, but many of them are actually just kind of parked in lymph nodes and in malt or in other lymphoid tissues throughout the body, okay? They're waiting to be activated by an antigen that they are specific for. When they do get activated, they differentiate into plasma cells and memory B cells. The plasma cells start to make antibodies. They turn into little factories making antibodies. And antibodies are not actually living cells. They're just little proteins that can go out, and they have special attachment sites on them that are adapted to specific antigens. Okay, so once an antigen has been met and it activates a plasma cell, many millions, thousands, whatever, hundreds of thousands is probably a better number there, of antibodies are going to be produced by those plasma cells. They will also divide and make new plasma cells as well that all produce the same kind of antibodies once that immune response is mounted. And we call that humoral mediated or antibody mediated immunity because it's not cellular. It's these antibodies that are released into the plasma, into the lymph, and those are what go out and bind to the antigens that have invaded. And once those antigens are bound, it marks them for things like natural killer cells and cytotoxic T cells to come along and destroy. Okay, natural killer cells make up about 5 to 10% of the circulating lymphocytes as well. Okay, so that's kind of an initial review of adaptive defenses. Now we're going to talk about some different ways of activating the adaptive defenses. So we have several different ways. We have forms of immunity that are part of our adaptive system. So we have these active immunity, and we have passive forms of immunity. Okay? Then on another metric, we have natural forms of immunity, and we have artificial or artificially acquired immunities. Okay, so an active natural immunity would be an infection, something that your body got involved in and actually had to fight off. Okay, so you got COVID, let's say, right? And your body fought it off. So you developed some natural active or active natural immunity to that infection. A passive natural form would be getting antibodies through breast milk. So certain types of antibodies can be provided to a baby while it's breastfeeding from its mother. So her antibodies, things that she's developed immunities to, will be given to the baby, and the baby will have passive natural immunity. Okay. Another form of artificially acquired but active immunity is when we get an active or an inactivated antigen in the form of a vaccine. Okay, so when we're vaccinated, they give us an inactivated form of an antigen that we develop an active immunity to. Okay, because this antigen got into us, it didn't make us sick, but our bodies recognize it as that's a bad guy, right? They put up a response to it, and we consequently are develop a more of an immune response to it. Okay, so artificially acquired active immunity is from these attenuated or inactivated antigens that are present in vaccines. A passive artificially acquired immunity is when we get an antibody. Our antibodies infused into us. Infusion kind of thing. So this happened during COVID. Remember how we talked about people who'd had COVID? They were asking them to donate plasma because that gave people who were not mounting an appropriate immune response additional antibodies to COVID so that they could help and fight the COVID off better because they got these antibodies from somebody else who'd already defeated the infection. Okay, so again, we have these various ways of getting our immunity activated and up and working. Passive immunity is through either naturally or artificially acquiring antibodies to a specific antigen. And active immunity can be acquired naturally or artificially by having the antigen itself introduced to our bodies and our bodies developing a response to it. Okay, so those are the four main ways that we can develop adaptive immunities. Okay, so a really important concept to understand in terms of how we develop immunity to such a huge variety of different antigens is clonal selection. Okay, so in the development of the T cells and the B cells, there is some factors where they become very specific, right? They learn how to be very specific, but there's also a lot of versatility. So the way we have both of these factors, specificity and versatility, is through this process of clonal selection. So we can produce, in theory, 10 to the 15 different types of antigen receptors. So a quadrillion different types of antigen receptors because of the huge potential for our genetic code to create different types of proteins, okay? And those different types of proteins create three-dimensional shapes here that can fit specific antigens. So the specific antigen would fit into this one, right? So we can create, when we're making our T cells and B cells, we can create up to, theoretically, we don't maybe actually do that many, but in theory, given how many different genetic possibilities there are and the recombination possibilities that we have as the T cells and B cells are developing, we have an almost infinite number here of potentials, right? So we create lots and lots and lots and lots and lots of T cells and B cells with different types of receptors in their developmental stages, okay? That is important because we need to have a ton of different potential matches for any type of antigen that could get into our bodies. And of course, the possibilities there are also really, really huge, right? There's a lot of different potential things that could get into our bodies and infect us. So we have to have antigen receptors for those things, or the possibility of antigen receptors for those things. So we're going to, bring this down to a manageable number. So let's say we have a T cell here that looks like this. We have a T cell here that has a receptor like this. We have a T cell in training here that has a receptor like this. And we have another one here that is has a little receptor like this. Okay, so in order for us to decide whether these guys are good to go in or out, like if we're going to let them out of the thymus or the bone marrow and into circulation, depending on if they're B cells or T cells, and into the body in order to do their job, they have to go through a process of clonal selection. And we need to find out if they have self-tolerance or not. So we make sure that they don't actually attack our own cells, right? So first of all, we have to rule out any one of these that attacks our own cells. So if our own antigens happen to look like this, and they would match this cell gets eliminated because it is going to harm self cells, right? So it gets eliminated. The others that are not going to hurt ourselves, they can move on to the next step, okay? However, once they're out there, the antigens themselves undergo clonal selection in terms of deciding which ones are going to actually replicate and become activated. So if we're exposed to an antigen, something gets into our bodies, and it has this antigen on it that matches this T cell, right? Or B cell, right? It's going to activate this one. Once this is activated, it's going to make a whole bunch of clones, okay? So this is what we call clonal selection. First of all, they undergo this process in the thymus and in the bone marrow to make sure that they're not going to damage our body before they're released. Once they're in circulation, or once they're out there in the lymphoid tissue and they meet an antigen that matches their unique combination of protein receptors, then they clone themselves. They become activated and they increase in number greatly. And then that helps them. If they're B cells, it helps them produce more antibodies for that antigen. If they're T cells, then they produce more T cells that are going to be able to fight. And they differentiate into the different types of T cells so they can mount a really well-balanced reaction to that antigen. So clonal selection is the process of deciding, first, we're not going to hurt ourselves. We'll eliminate the bad guys that are, the types of T cells or B cells that are developed that might accidentally hurt us, right? And then once they're out in circulation, whatever antigen they run into, if they match that antigen, right, then they're going to clone, replicate themselves, and mount a really strong immune response. Then the last step for that is the memory cells, right, that can occur. So if we cloned, we mounted a response, and we fought off that antigen, we also retain a memory, right? So the cells that were activated, more of them hang around and are ready the next time that antigen comes in. So we have a stronger secondary response to that antigen if we're ever exposed to it again because of this property of the immune system, this memory cells that are ready. So initially there's only maybe a few of these T cells out there, right? But after that exposure, now there's lots of these T cells that become memory cells and are ready to fight in the next encounter. Okay, now we're going to be taking a deep dive into the weeds kind of thing on how these cells actually all work together and how they accomplish their immune responses. So we're going to have to be introduced to a new concept here, which is called the major histocompatibility complex. So here is an antigen, right? Here is a antigen. This is a bacteria that got into our bodies, okay? And it has these antigen fragments that are on its surface, okay? So the whole thing or its fragments will be recognized by the body as an antigen, okay? Here's a normal cell. The normal cell also has little markers on its surface. These markers are class 1 major histocompatibility complexes, or MHCs. Histo means tissue, so a large, which is where the major comes from, a large protein on the surface of the tissues that marks it as being a self tissue is these MHCs, right? So the MHCs mark it and say, hey, I'm a normal tissue. I am compatible. I play nicely with the rest of you guys because I'm part of the same body. So a class 1 MHC indicates that this cell is a self cell. Okay, we don't want to hurt it. Here is what we call an antigen presenting cell, or a macrophage, or something else like a dendrite or a dendritic cell, something along those lines. Okay, so multiple different types of immune cells that are part of the innate immune system can act as antigen presenting cells. So when they meet up with this antigen here, this bacteria, they're going to engulf it, and they're going to take it inside to their cell. They're going to digest it. As they do so, they're going to break down its little proteins, and they're going to mount those little antigen proteins on their own surface, okay, as they digest it. And then they're going to take little pieces of it, and they're going to say, hey, look what I caught, okay? And they're going to use their type 2 or class 2 major histocompatibility complexes on their surfaces. So only the APCs, or the antigen presenting cells, have these major histocompatibility complexes, these class 2 types. And when they destroy an antigen, they display trophies, essentially, of they got that bacteria. And they put them on their MHC2s along with their MHC1s that they also have, which says, I am also part of the normal body here. But then they also present these antigens to other cells in the body. Okay, so when they present these to the other cells may be a T cell. Okay, so let's say it's a T cell. We're going to kind of follow it down that rabbit hole. So when the T cell that has this very specific antigen receptor on its surface that matches, as you can see, these little antigens on the antigen presenting cell, it's going to be able to bind there. Also, if it binds to a bacteria here, itself directly, right, it's going to be co-activated. So this was an MHC1 with the antigen for the bacteria, okay? And this is an MHC2 with the presenting cell there. And so this is saying that the T cell found a match, and then an antigen presenting cell said, yeah, I already got somebody like that. This is co-stimulation happening, and this T cell is going to get activated because it knows it's run into the bad guy itself, and the antigen presenting cell agrees with it. It says, hey, yeah, that was a bad guy, and so we're going to activate a formerly inactivated T cell. Those activated T cells undergo clonal activation. They start to clone themselves. They form cytotoxic T cells. Okay, they form memory T cells, they form helper T cells, and they form regulatory T cells. And the T cells have on their surfaces, they have something called CD markers, or cluster of differentiation markers, and that helps to identify which type of T cell they are. Okay, so there's 350 different types of cluster of differentiation markers, but we're going to look at just a few of them that we understand fairly well and show how those work. So cytotoxic T cells, or CD8 markers, are displayed on them. Okay, the CD8 cluster of differentiation marks them as cytotoxic T cells. Okay, CD8s also are found on regulatory T cells. Okay, helper T cells, on the other hand, have CD4 markers. Okay, and they respond to the MHC class 2 bindings, so that they'll be able to then bind with these antigen presenting cells. Cytotoxic T cells will be able to bind directly with the antigen. So these activated T cells, cytotoxic T cells, are going to be able to directly fight these MHC1 complexes with their CD8s. So their CD8 is going to match the antigen now on an MHC1 type 1 or class 1 complex, which would be right directly on the antigen. So cytotoxic T cells then are going to be able to recognize specifically the bad guys, okay, directly. And then they're going to undergo or engage in cell-mediated immunity. So they're going to then directly attack, essentially, the antigen cell, okay? And to do that, they're going to destroy it by releasing perforins. Okay, perforins are proteins that will, like we talked about before, they'll create a little hole in that bacteria and it'll destroy it. They can release cytokines that will activate genes inside the bacteria, which will tell it to kill itself. Okay, kind of like a nasty social media post, and it will self-destroy or apoptose. Okay, so apoptosis is one way that the CD8 cytotoxic T cells can cause the bacteria to die. And then the last thing that they may do is create a lymphotoxin that could kill it by creating a really hostile environment around it. Okay, so CD8 cells, like I said, when they're activated, okay, from this originally activated T cell, we create cytotoxic T cells that respond to MHC type 1 complexes on the antigen itself and engage in cell-mediated immunity or cell-to-cell combat. Okay, the CD4 cells, or the helper T cells, we'll talk about more. They don't directly fight the antigen themselves because they recognize the major histocompatibility complex 2 on antigen-presenting cells, and they work with B cells as well. So we're going to talk about them a little bit more when we get to the B cell concepts a little bit more. But in the meantime, we know that helper T cells are also activated when this original T cell is activated. Memory T cells are those that are going to stick around for years to come and be ready for the next time that this antigen dares come into our body. And regulatory T cells have a couple of different functions. So the regulatory T cells, as I was saying, those secrete suppressant factors, or in other words, they slow the reaction down. They said, let's not go overboard here. So the regulatory T cells have a kind of inhibitory effect on the T and the B cells that are involved in this specific antigen attack. And they sort of, like their response is a little delayed. So if the other cells are peaked there, the regulatory T cells are going to come along and peak a little bit later on in the process. So they don't start right off, but they do start to grow as the response continues. So they come in as a regulator toward the end of the initial effect, and that they have to limit the immune reaction to a single stimulus. So they act as a safeguard to prevent an overreaction in the case of an antigen invasion. The last thing I wanted to mention is that we have these CD3 cluster of differentiation cells that are present on all T cells. So cytotoxic, memory, helper, and regulatory T cells all have the CD3 markers. Okay. So whenever you have an immune response, it involves a lot of cell-to-cell communication. And cytokines are one of the ways that cells do this. So they release chemical signals that communicate with each other. So within an immune response, we release a lot of what we call cytokines. There are 6 major types of cytokines. So we have interleukins, interferons, tumor necrosis factors, and phagocyte activating chemicals. And we have colony stimulating factors. And then we kind of have a miscellaneous catch-all cytokines. So we have these 6 main categories, and we're going to kind of break this down a little bit and talk about each one of them individually. So interleukins have a broad range of effects. They can increase T cell activity, particularly their sensitivity to different antigens on macrophage membranes. So their sensitivity to antigen presenting cells. So they're going to notice these guys a little bit more when interleukins are around. They can increase B cell activity in forming plasma cells and antibody production. So increased plasma cells and increased antibodies, B cells. It also enhances innate immune activity, so inflammation goes up, mast cells, which are part of the immune response, releasing histamine and such. It increases adrenocorticotropic hormone production. It increases scar formation, which is useful for healing initially, and it can increase body temperature. So this is part of the innate immune response that interleukins support. And they can also, a fourth function here is suppressing immune function. So different interleukins can actually also have an inhibitory effect if things are getting out of hand. So interleukins have stimulating effects on both the innate immunity, the adaptive immunity, and also a regulatory effect on the immune response in general. So some specific interleukins are interleukin 1 and interleukin 2, okay? And these are really important in stimulating and maintaining the immune responses. When they're released by activated macrophages, so remember macrophages have run into an antigen and eaten it, they're activated, they're going to release these guys, okay? And when released by these activated macrophages or lymphocytes, these cytokines are going to then stimulate the immune cells of other secreting cells. So it's going to just kind of ramp up this whole process. This result in a positive feedback loop, okay, that just recruits more and more immune cells. So interleukin 1, interleukin 2, released by macrophages, gets more lymphocytes involved and amplifies this entire process. We talked about interferons earlier, so we're going to skip over to tumor necrosis factor. Tumor necrosis factor slows, as you can imagine, the growth of tumors. So that's important. We want them to slow that down, right? It can also kill sensitive tumor cells because tumor cells are very fast growing. So they're very delicate, if you will. They're more easily killed than normal mature cells. It helps to stimulate leukocyte production. So increased white blood cell production, particularly the eosinophils, which can create fever and T cell sensitivity to interleukins. So the activity of these white blood cells is stimulated. It helps to activate macrophages, which we want to be activated against the tumors. And the macrophages can then secrete tumor necrosis factor themselves and carry those molecules on their presenting membranes, or like on their major histocompatibility complexes, right? They're going to display the tumor factors. And then cytotoxic T cells. T cells, remember, these guys do cell-to-cell combat, okay? And they also produce a tumor necrosis factor, okay? So they can help activate a response against tumor. So this is the way our body can respond to cancers and abnormal cells in our own body by releasing these cytokines. TNF, or tumor necrosis factor, helps to slow those tumors down, increases the activity of the white blood cells and the macrophages and the cytotoxic T cells. So all of them can kind of be, activated and stimulated to attack these tumor cells. So the phagocytic activating chemicals are kind of self-explanatory. They attract macrophages. They help to kind of coordinate the immune defense response by activating specific macrophages. And so they attract both micro and macrophages, such as the neutrophils and the monocytes, some of those smaller ones, as well as the larger macrophages. And they also prevent them from leaving the infection site too quickly. So they attract them and then kind of keep them hanging out there a little bit longer. So those are important cytokines for maintaining the response that's needed to get rid of the infection. And then we have colony stimulating factors that we mentioned earlier. Colony stimulating factors. And these are important because once T cells are activated, active T cells, and monocytic macrophage systems and endothelial cells and fibroblasts, all of these are stimulated to move on. So here, monocyte and macrophages and endothelial cells, which are important for healing, right? All of these, oh, and also fibroblasts, of course. Fibroblasts, which are going to help create the scar tissue and such. These are all going to be activated. So it's basically saying, hey guys, we need to go into hyperdrive here. We need to get rid of this infection. We need to heal ourselves. And that's what colony stimulating factors do as cytokines. They also stimulate the production of RBCs, which is a good thing in the bone marrow, and white blood cells as well in the lymphoid tissues and the lymphoid organs. So colony stimulating factors, which we've talked about them sort of individually before, but as a response to infection, they are produced as cytokines, and they activate a lot of different systems that are all involved with healing. Okay. Another way to classify cytokines is by their origins, and so you have the lymphokines are coming from lymphocytes, right? You have monokines, and those are coming from monocytes. And macrophages and other antigen-presenting cells. Okay, monokines can be released by those. And lymphocytes and macrophages also secrete other types of cytokines, so many types of cytokines. Again, it's a very complex system, all right? And so they have to have lots of different messaging systems and ways of communicating chemically with the cells around them so that the response is just strong enough to get rid of the infection but not cause damage to our own tissues. So cells involved in this adaptive immunity and tissue repair can also secrete cytokines. All of these other, even the other tissue cells, can talk back and say, hey, this is okay, this is not okay, etc. So the entire immune response is one cell communicating to this cell, communicating back to that cell. There's all this interplay between the cells. So the cells have this cyclic way of communicating with each other. Sorry, I'm trying to show this as two-way communication between the cells, all the different types of cells that are involved in the immune response. Okay, so it's really important that all of this stays in balance and that the cells read each other's body language, so to speak, and they get those communications correct and the responses are appropriate throughout the entire immune reaction. So specifically in terms of T cells, the functions of the cytokines help to increase T cell differentiation and multiplication. They help to accelerate the cytotoxic T cell maturation. So both helper T cells and cytotoxic T cells are going to be stimulated. It helps to attract macrophage, or them to be attracted to the macrophages, and it attracts the stimulation and the activity of the cytotoxic T cells. And it promotes, of course, activation of the B cells as well. Okay, so this communication, T cells to B cells to macrophages to other tissue cells, all of that is facilitated by cytokines.